Good evening to all. Welcome to 5 days National Level Faculty Development Program on Digital Transformation and Innovation in Biological Sciences, redefining the role of two key teachers. Dear participants, kindly make note of the instructions to be followed for the uninterrupted and smooth going of the webinar. Kindly mute your audio and video during presentation. Kindly ensure high-speed internet connection for uninterrupted listening. To enhance good audio throughout your meeting, please put on your headset. Only the host will present. Others, please do not present at the time of meeting. Please share your questions and feedback in the chat window, which will be addressed at the end of the session. Kindly do not unmute to raise your questions. Complete attendance throughout the FDP will be appreciated for the issue of e-certificate. Shall I start, ma'am? Ma'am, shall I start? Yes, ma'am. Good evening, ma'am.
Good evening, Trichy.
Good evening, ma'am. Yes, ma'am, shall we start, ma'am? Ma'am. Shall I start, ma'am?
You can start, Dema. She's not here.
Okay, ma'am. Okay, ma'am. Okay. Thank you, ma'am. Good evening to all. I am Dr. D. Prema, Assistant Professor, Department of Biochemistry, Holy Cross College. It's my pleasure to be a moderator for second day faculty development program on virtual lab on HPLC and mass spectrophotometer workflow for drug analysis. On behalf of Biochemistry Department, Holy Cross College, I would like to extend a very warm welcome to all of you. First of all, let's thank the Almighty God who giving us the mercy and blessing until we can attend in this event without any obstacles and also in this happy place and time. Virtual lab refers to a virtual teaching and learning environment aimed at developing students' laboratory skills. Virtual labs are excellent digital resources that help to save cost and enhances students' productivity manifold. Before the presentation begins, I would like to introduce the Honorable Speaker, Dr. S. Girija, Associate Professor, Department of Biotechnology, Bharathiar University, Coimbatore. Ma'am has received her PhD from Bharathiar University in the year of 2001 on in vitro culture and selection of sodium chloride tolerant cell line in Mumbai. She is having 19 years of experience in research and 16 years of experience in teaching. Ma'am has guided 12 PhD and MPhil students. She has awarded INSA Course of International Award in the year of 2011 to 12. She has visited to South Korea for bilateral exchange program. She has served as member in various committees in various universities and colleges. She has organized many national and international seminars, workshops, refresher courses, and orientation programs. Ma'am has acted as both principal and co-principal investigators of various funded projects sponsored by UGC, DRDO NMAS, DRDO FRL, DST SERV, RUSA, and DBT. She has deposited many gene sequences in World Gene Bank. She has published more than 50 national and international papers in reputed journals. She has attended many seminars, conferences, workshops, and oriented program conducted by universities and colleges. She has also acted as a resource person in various seminars, symposium, conferences, and other external activities. Ma'am is an expert in HPLC and mass spectrophotometry analysis. I feel so proud to have such an eminent scholar as a resource person for today's session, ma'am. Dear participants, kindly join with me in welcoming ma'am. Ma'am, welcome, ma'am.
Thank you, Prema. Thank you.
Now I hand over the session to ma'am. Ma'am, the session is yours, ma'am.
Yes, Thank you, Prema. Thank you for your nice introduction about me. And I'm very happy to see almost about some 50 plus participants who are attending here. And for me also, this is a 2 hours, first 2 hours program in online. Usually only Viva will go up to 2 hours or 2 hours. Otherwise, our talk will be for 1 hour only. It's the first time that we are planning for 2 hours. Actually, if it is a working time, like if it is a daytime, and me and Prema have planned to do an online demonstration from my research laboratory, like how we can do a HPLC analysis. And we have expertise and we have facilities for doing a HPLC analysis, and we have been working for more than 15 years. So I thought I can share some of my research as well as how we can do the drug analysis using the HPLC. And I will also touch some workflow in mass spectrometry and how the mass spectrometry can be used to analyze your drugs. So now I'll present my slide accordingly. In between, if you have any doubts and queries, definitely you can stop, because this is going to be a 2 hours program. Better you can interrupt me and interact with me at any time. You can stop. This is going to be a mostly towards the practicals, because now in this situation, the theoretical paper is easy for us to handle. Only thing we face problem is in the practicals. Hope you all agree with me. And I'm also happy to see all of you to spend time to inculcate your knowledge in this time, because this is all your. Is my slide is visible? Any
Yes, ma'am.
moderator? Any moderator? Or
Yes,
Prema?
ma'am.
Or Prema will act like as a moderator? Any moderator?
Yes, ma'am. Prema ma'am is today's moderator, ma'am.
Okay, then that is fine. Okay, because,
Okay, ma'am.
because I in
It's
between.
ma'am.
Yes.
Ma'am, your slide is visible, and one more thing, ma'am. Students, participants will post their queries at the end of the session, ma'am. Maybe you can speak for one
No,
hour, one and a half hours, and
no.
maybe the questioning session can be followed by that, ma'am.
Okay, no point.
Thank you.
That's okay. So whether my slide is visible, like so,
Yes, ma'am, it is visible.
because you all of you are a teacher, I'm also a teacher. So don't differentiate me that I am a speaker, and we all here to discuss about the research, one kind of on one topic, how we can go ahead with this research, and we are all sailing in the same boat. As we have put up in the university, and we have established some laboratories, and getting the funding from the various funding agencies. So we have some workflow structures facility in our department. So we all can do, we can think like how we can solve the practical, how inculcating the practical knowledge to the faculties as well as the students. So today's my talk is a HPLC, and first of all, I would like to thank the Holy Cross admin for inviting me for this conference of FDP programs to interact with you people. I'm happy to share my experience. So once again, I thank the Holy Cross admin for inviting me, and also the mediators, Dr. Prema, for requesting me to present for the today's FTP program. Okay, so now we'll get into the topic. So HPLC and mass spectrometry workflow for drug analysis. You may think that what is the necessity of the title and why? Because most of you might have done this either as a part of your PhD work or as a part of your students project. Okay, so I don't know how many of you having a hands-on experience in this HPLC. And mass spectrometry, definitely it is not possible, because even in Bharathiar University, we recently purchased a LCMS, and I'm now processing a GC mass with the thermal desorption unit. And hopefully most of the institute now has the HPLC instrument. Okay. So, but thing is, we may not be knowing how we can use this instrument and what are all the different purposes can be solved by using the HPLC. Okay, so Prema has requested, like, ma'am, you can also talk something about your COVID drug, possible drug for the COVID, because the present situation we are facing the problem because of the SARS-CoV virus. Still, we are not able to figure out a single molecule which can act as an antiviral compound to kill the viruses. And before entering, this is the topic which really attracted me: the digital transformation and innovation in biological sciences, redefining the role of 2K teachers. So this 2K teachers, this word is really impressed me. Like we are really a different teachers, right? Okay, so still last 2018 and 19, we were with the chalk and talk, and we were facing with the students directly in the life was so good. And we can threaten the people, we can walk. If you are not able to complete the syllabus today, we can postpone it to tomorrow. And the practical session, we have a different set of experiment. So life was okay. Now it is completely transferred into the digital media. Okay, but still some of the things are really comfortable, like you can focusedly sit on the slide. Even before that, my style of teaching was I never used chalk and talk. I always depend on the PPTs. I make very beautiful presentations. Even I too created YouTube channels where I post my all PPTs as a practical knowledge, because I don't want to take much on the concentrate on the theory. Theory anybody can study. Okay, if you Google it, whatever you want, Google will give you all the answers. Now all the research paper have been published. Even after COVID, many of the papers, important journals, which is a paid journals, now they have become open access journals, because they want the papers to be available for all the researchers. Okay, so now I really appreciate for this topic, which really impressed me, the role of 2K teachers. Definitely, this will become a history, like how we faced teaching, because for others that is okay, but we are teachers, are we control thousands and millions of youngsters on our hand, and we are controlling, and we are inculcating their knowledge. We are keeping them and continuously engaging them in the science or any subject. So hope we never go down. We'll always grow. So whatever the situation comes, we are this taught us, like we can cope up with any of the situation come. We never stop our teaching. Okay, so I salute all the teachers. I'm also happy to be a teacher. Whatever maybe, whether we are earning money or not, that is not secondary, and we feel satisfied inside that we are teaching, we are transferring our knowledge to the young kids. So this is our university. This is the Bharathiar University. Some of you might have visited. And so we have a very beautiful complex, because this is in the west foothills of Western Ghats. So the Mardamalai is just 2 kilometers from here. So we surrounded by hill stations and hill area. Climate will always in a very nominal climate. You never had get the hot climates. So we have various departments in our campus, including especially the life sciences are very strong, and we are the biotechnology department is a very good department. We have all the recent facilities. Once if any the situation is over, I welcome all you to visit our department. And so this university was named after the Bharathiar. He is a very great poet. Like so, he fought for all the women freedoms. all his words are very sharp towards giving a freedom for the woman, and we have a very good convention centers, all the facility. It's a very nice campus to visit. And this is our department building. Actually, I don't I because the file has become very large if I put so much of photographs. And this is our department. Like you have 3 floors in the only for biotechnology, and we have a very good facility inside. All we have the research areas of different discipline like plant biotechnology, animal biotechnology, microbial biotechnology, and the bioinformatics. And we have all facilities like animal cell culture, transgenic facilities. We have containment house, we have animal house, separate animal house, all the facilities. When we came in 2003, nothing was there. We all worked together and got the funding from different agencies, and we elevated to this level. So now we'll directly enter into the topic like a drug discovery. Everybody will talk about drug discovery. Okay, so now even I was thinking like, see, we are in India, we have a very less facility, and we have a limited facility, we have a limited funding. Our country is like this, so we are not able to, we cannot work like the foreigners, because they people in University of Boston, people in University of Harvard, people in University of California and Canada, they have all such kind of facilities. If any problem, any disaster come to the world, they will solve the problem. Like that, even I was having a convention in my mind. Okay, so but all this were vanished for last 2 years. Even in spite of having all the facilities, till now they are not able to figure out a single molecule for treating the SARS-CoV virus. Okay, so but still, I will not criticize. What I am trying to tell here, we are no way inferior to them. We are more knowledge, and we have all facilities in India, and maybe some of the facilities may not be with us in our institute, but we have in other higher institution. We can collaborate with them, and we can find out any drug or any research problem. That is my kind of opinion. Like, hope you all also will agree with this. So coming to the development of a new drug, so it's not easy. As everyone say, drug discovery is not easy, but I feel that it is not that much difficult as yearly. When we were doing a PhD in 99 and 98, and it was very difficult, because when I was doing my PhD, I never seen a HPLC, and I never get into any higher end facilities. But nowadays, every institute is affordable to buy all these instruments. Only thing we need is your lack of hands-on training. Okay, so instrument will not solve all the problem. Only thing you should have a hands-on training. And also our students, why they are not able to place in any industry, because of the lack of practical knowledge. Okay, so as a faculty, we have the responsibility to learn the techniques on ourselves first. Then only we can transfer our knowledge to the students. Okay, so hope most of you as a teacher, you will be handling the practicals and all basic experiments. But I wish you all to get trained in the high-tech instruments like DNA sequencing, mass spectrometry, and microscopies. Nowadays, everybody is offering a facility training. Even in online, many of the training, we have a DNA sequencer in our institution. Even they have conducted a 1 week online training program for the sequencing and everything. Okay, so that's what my apprehension, that we all should get a practical knowledge. So now the drug discovery involved the identification of new chemical entities. Okay, so new chemical, we have to identify a new chemical entities. They have the required characteristic of druggability. So we can identify many new compounds. That's not a very big task. But the thing is, it should have the characteristic of the druggability, because acting as a drug, the drug should have some of the properties. Okay, Hope some of you might have studied the pharmaceutical biotechnology, where if you define a drug, what is the drug? The drug should have some properties. Okay. So, and in medicinal concept, these entities can be sourced either through chemical synthesis or through isolation from the natural product. So, some of the drugs are really breakthrough. So, still we don't have the alternative for the paracetamol. Okay. So, hope you all agree. Paracetamol is one of the antipyretic agent. So still now we have the modification of paracetamol, and we don't want to replace it also because it doesn't cause any toxic. Maybe some of the work they publish continuous consumption of paracetamol is creating that toxic disorder. So all that is a part of a toxicologist's work. So like that, some drugs we don't want the alternative. But what some of the unaddressed diseases like cancers, Alzheimer's, now we see the viral diseases, we don't have a drugs. So what is a drug? What is a drug? Is it only from the plant? It's not like that. Okay. So any small organic molecule can act as a drug, and the protein molecule can act as a drug, and peptides can act as a drug. Now, and if you take United States of India, many of the professors they will have a peptide industry. So now, because peptide, it is the protein with the less than 24 amino acids. We call it as the peptides because it is a very small molecular weight proteins. It is easy to act as a drug. So many antimicrobial peptides, even for the cancer nowadays, many peptides are targeted. So this is produced in a larger scale by using, mostly in US they have many peptide drug industries. And other drugs is a vaccine. So vaccine we all know because we all are administrating the vaccine. And in vaccine you have a different types. Okay, so now you would have heard about the different terminology like Covaxin, Covishield, and Pfizer and things. Only the technology will differ. If you take a Covaxin, this is a whole virus is unattenuated. Attenuated whole viruses is injected as a. They grow this as in the animal media by using serum, and then they will kill the virus. And this was a traditional technology what Edward Jenner, what we traditionally use for production of a vaccine. But whereas when you take a Covishield, that is a subunit vaccines. Okay, subunit. Hope you might have taken the classes for MSc for immunology. If any of you are teaching an immunology, you might have taken the classes on different types of vaccines. And this Covishield is a subunit vaccine where they have designed only based upon the spike protein. Okay, so it will produce the antibodies against only the spike protein because that protein is essential for the infection into the human cell. Okay, similarly we have other vaccines like Pfizer and all. Now the mRNA technology has come. Now it is ruling the world. Okay, it is ruling the next generation, and the next century will be only people who will work on the mRNA because mRNA is directly translated into the protein. So nobody will work on the DNA. They all will started working on mRNA. That is what the Pfizer vaccines are, mRNA based vaccines. And even in the plant, when you do a transgenic, we clone the mRNA. So mRNA is a very good candidate for the molecular biological work. So this is a vaccine. And many of the recombinant proteins are acting as drugs. To start with your insulin. Okay, so insulin is a recombinant protein. So when you see earlier days, how we get the insulin, they slaughter and cut the animals and they will isolate the insulin from the urine or the blood samples. So it was very difficult, right? So once they identified the gene which code for the insulin in the human, this was synthetically synthesized, then transferred to the bacteria. Now we have a recombinant insulin. Okay, that's what we are able to get the insulin for a cheaper price. You will get for 50 rupees, you will get for 150 rupees, you will get for 500 rupees. Why this gradation is there? Because Insulin can be produced in a bacteria, can be produced in an animal cell culture, can be produced by using viruses. So based upon the production system, the cost of insulin is also vary. And monoclonal and the monoclonal antibodies. Okay, so that's what people wonder. Even in the United States of America, they have the facilities. Within a 6 hour, they can synthesize to any monoclonal antibodies. So antibodies are the best drug. Even in recently there are 11 and 14 antibody drugs were approved by the FDA. Okay, so now you had heard about that even in the COVID, people will transfer the plasma because that plasma will have your antibodies. So monoclonal antibodies are an efficient drug. And coming to the natural product. So maybe if you are a chemist, you can go for small organic molecule synthesis. If you are a biotechnology, if you have a facilities, you can go for a protein and peptide. And vaccines, you need to have biosafety level 3 labs. And even still you can collaborate with any lab which work on the viral research. And coming to the natural product, so we are comfortable with the research of the natural product. Okay, so most of us like they work on the natural product because this is feasible and this is easy. And also the main reason is India has a number of potential herbal plants which with the very potential therapeutic chemical entities. Okay, so that's what. So this is the kind of drug. So in that you have to choose which drug you are going to work. Everything is possible. Recombinant DNA protein. It's not a big task. Okay, so we have all the facilities. Even every year we clone a different genes because my expertise is only on the recombinant technology because we clone the genes, we do metabolic engineering and also the genetic transformation to increase any pharmaceutically important product in the either in the food crops or the. So that is a different story that we'll talk on some other day. So okay. So in 2008, if you see the FDA approval, it's a 59 new drugs, have 42 new chemical entities, and 17 biological were approved. Among this, 11 is the monoclonal antibodies. This is approved, which account for almost 20% of all the drugs in 2018. Okay. So now hope you got an idea which drug you want to work it out. So this is a percentage of drug approved by the FDA in 2018. Like you can see, 2% inorganic. A natural product, there are only 16% of natural product, and many of the fluorinated drugs and the pegylated enzymes. Now people are concentrating on pegylated drugs. So pegylated means attaching the peg molecule to any drug or the protein or the peptide. Now because this is a very it will easily pass through the cell membrane. Now people are interested in the pegylated drugs and monoclonal antibodies and other small molecules and combinational drugs were approved. Okay, now we'll come to the natural sources. What are the main drugs which are approved by the natural sources? Okay, so the first commercial pure natural product introduced for therapeutic use is a morphine. Okay, so morphine, it is an analgesic drug which is acting as a painkiller during your surgery or any unwanted body situation. So this was in 1920. Actually, this morphine is also a drug. Okay, it's also a drug they will consume for other drugs. And then the first semi-synthetic pure drug aspirin is produced from the salicylic sulfur introduced by Bayer company. This is a very famous company in 1898. Actually, it is missing. And followed by a cocaine drugs and also and the digitoxin, which is a cardioglycosides used for the treatment of heart problems, and quinine, the anti-malarial, the hydroxychloroquine. Nowadays, even this has now attracted the COVID patient. Many of them, they consume the hydroxychloroquine because to kill the SARS-CoV virus, and the pilocarpine eye drops were discovered, and the Taxol. Okay, so this is now the breast cancer is completely curable. Okay, because of the Paxlovid. They isolated the Taxus brevifolia plant, and they modified the groups, and now they attached with the DMAE polymer. This is a pegylated Taxol is now used for treating the breast cancer, and they are targeting for the receptor proteins. So breast cancer is curable only because of the God shape, because cancer we know. So you don't have a specific marker for the cancer, because normal cell and cancer cell will look like the same. Hence, it is very difficult to target the drug for the cancer cells. That's what people suffering from the cancer will. If you take the drugs, these drugs will affect the normal cell also. That's what we are not able to cure them. But whereas the breast cancer is completely curable, because they have identified the receptor proteins, BRCA1, BRCA2 receptor proteins. So this Taxol, they modified the BRCA receptor protein, so they targeted directly to the breast cancer. Then the artemisinin from the traditional Chinese plant Artemisia annua, and for the malaria and silymarin extracted. So in this list you can find, if you go to the FDA website, you can find some more list of the drugs which is approved. So this was Taxol. This is a potential drug for the cancer identified in 1967 from the bark of Pacific yew tree, Taxus brevifolia, by the American Indian for the treatment of inflammation. Wherever any research is there, there will be an Indian will be there, Indian brain will be there to identify any of the I5 research. So they isolate from the bark of this Taxus plant. So now we'll come to what are the possible drug targets and potential treatment for the COVID. So COVID, this SARS-CoV virus, this is the structure of the virus. Hope you would have seen in many of the Google search. And this is a RNA virus. Okay, this is a RNA virus where you could see like, and it has a virus and it is covered by the protein. And this is the spike protein. This is the spike protein. So this is what they synthesized the antibody coding genes for this spike protein for the vaccine production, and they also is a potential target for like how we target for the BRCA receptor. We can also in future we can design the drug to target for this spike protein, and it has a nuclear envelope, and it has a nuclear protein and the membrane protein, and this is the structure of your SARS-CoV. Okay. So why it is called SARS-CoV-2? Because the sequence of the SARS and the COVID is 80%, which has a sequential similarity. So that is what this is named as SARS-CoV-2. So what are the drug targets and potential treatments available? See, globally there is hard research are going on. So many call for is coming in DPT. If you go to a DPT website, now international collaborations have been called for to work together with an Indian scientist, because they suspect we may have some drug entities in our country. So now they are asking for the collaboration. Many countries are asking for the collaboration with us. So here there are global efforts are already way underway or footing to identify the best drug combination to address the diseases. To achieve this, it is essential to first identify the drug. So isolating compound is easy. Okay, you have to identify the target for which molecule of virus you are going to target. So we have seen, that's what I shown the viral structures in the early slide. Okay, so we have seen the structure, like we have a spike protein, we have a membrane protein, we have now the complete sequence genome is also sequenced. Even if it is mutated, there will be only 20% of the genes will get mutated, not 100% HF sequence difference will be there. Okay. So for that, we have to identify for the drug discovery. If you want to work on your drug discovery, you have to first understand the disease biology very well. Okay, so what is the disease which you are working? So now, if you want to target for a COVID, so now you have to understand completely the molecular mechanism of the COVID virus. Okay, SARS-CoV-2 virus. What is the structure? What is the molecular mechanism in which it is attaching to the human cell? How it is multiplying, and what are the things? What can be prevented? Like that, you have to work. First, you have to read. You have to thoroughly read about the biology of the disease. Then only you can go for identifying the compound. That compound you can target to the cell. Okay. So this SARS-CoV, I told you why it is called SARS-CoV, because it has 80% sequence similarity as that of the SARS. So it is named as a SARS-CoV-2. So this is a positive sense. So you, as a teacher, you might be knowing the classification of viruses. So the virus may be either of a DNA virus or the RNA virus, or a single-stranded virus or a double-stranded virus. Here, this SARS-CoV is a positive sense, single-stranded RNA virus with a genome size of only 30 kb size, which encode for only 2 ORF. See the size, but they say it is the very biggest size of the virus. But still, when we compare to our human and other eukaryotic organisms, this is just imagine how many ORF we are having, but this is simply has only 2 open reading frame. But still, it is not able to diagnose the replication, arresting the replication of the virus. So, what is the potential target? This is based on the literature, the knowledge which I have. So here, the spike protein is the forefront of infection. I told you, you can design the drug, which likely you can degrade the spike protein and interact with the ACE2 receptor. This is how they say why this COVID is not affecting the children, because this ACE2 receptors is absent in the children. That's what it is not affecting the children. Like there are many reports they say, like I don't, because we are not worked on these, only based on the literature and talking this. So this receptor on the host cells are thereby promoting the viral cell membrane fusion during the initiation of viral infection. So you can design the drug to target the spike protein, or you can target to the ACE2 receptors. Okay. Then inside the cell, the membrane and the E proteins is the RNA, which is guarded by the nucleocapsid protein. The SARS-CoV transmission, now, idla vandu romba detail na poporadilla, only just few thing I'll tell, because we have to see the other parts. So SARS-CoV-2 transmission is facilitated by the respiratory secretion, and now the person coming, recent report says that this also transmitted through stool, urine, and respiratory secretion. So this is the molecular mechanisms they proposed for the life cycle of your SARS-CoV virus. Okay, so it follow the endocytosis. So cell molecular biology எடுக்கிற டீச்சர்ஸ் வந்து எடுத்திருப்பீங்க, லைக் வாட் இஸ் எக்ஸோசைட்டோசிஸ் அண்ட் எண்டோசைட்டோசிஸ். ஸோ இட் ஃபாலோ தி, ஸோ இங்கே பார்த்தீங்கன்னா லைக் ஹவு ஃப்ரம் தி அவுட் சைட் it attaches to the membrane of your human cell. So here the receptor, the ACE2 receptors, and it attaches to the receptors, and it will come inside, and it releases viral genomic RNA, and this RNA will use the machinery of the human RNA machinery. Okay, so it cannot replicate itself. So this will use the machinery of the human, and it will replicate inside. So it will replicate inside and transcribed, and this produces the more number of viral RNA and viral proteins, and it will be transported to the nucleus, and also it will get a new regenerate as a new viruses, and it will go outside and affect all the cells. So this is the mechanisms still now available for the SARS-CoV-2. So in this you have to understand, like, so this is only a clumsy idea I told, but you have a very detailed molecular biology. What are the different types of a receptor, what is the sequences, and how we can target it. And now in the RNA-dependent RNA polymerases, also they are proposed as a drug. So similarly, you have to work on the disease biology, then only you can go for a drug discovery. So what are the drugs nowadays trialed against it? So I can't say that these drugs are used for COVID. These are trialed against COVID. So if it is possible, they will get cured. So the one of the main drug is target of for the RNA polymerase is a remdesivir. So you would have seen in many newspapers. Some of you might have taken this. This is the target the RNA polymerase of the SARS-CoV virus. Okay. And it has already been used for Ebola virus, influenza, hepatitis virus, and HIV virus. But nowadays it is used for this SARS-CoV virus. And also the other 3 CL proteases and an inhibitor of virus entry inhibitors, etamprazole, are used. And the inhibition of endocytosis, chloromycin. These are all. See here, you could see the chloroquinone, the hydroxychloroquinone. So it inhibits the endocytosis and acidification of endosome, inhibition of entry of the kinase inhibitor. This is a mechanism of this hydroxychloroquine. So similarly, many drugs are trialed but not approved by any agencies for the especially for this one. So now we will come to our research. Okay, what India is doing. India is taking so many steps. So Indians want us actually. We எல்லா DPT, DST எல்லாமே வந்து என்ன பண்றாங்க, now they want us to work on this antiviral compounds. Okay, antiviral compounds. So because whether my voice is audible?
Yes, ma'am. Yes, ma'am. Your voice is audible.
Yes, ma'am. It's clear, ma'am.
Okay, okay. So also now all our DPT and DST, they come for the special calls for the antiviral any targeted drug for the COVID. Okay, so DPT recently released a medium that prepared by inviting many scientists in northeastern England regions, and they listed many of the plants which has an antiviral compound. The preliminary work has been done previously, and this has been listed by the DPT, and they expect some of us to work. I think this call is still on. You can go and browse the DPT website. The last date is till 2022 is there. You can apply if possible. And these are all the various plant species, because I don't have much knowledge on the taxonomy, but maybe it is not utilized. Sorry. And so these are all the different. So whatever I shown in the previous in this compendium, and a complete book has been released. This is available in the DPT website, and then you can browse it. There are a lot of information on who are all done the work. Everything is available. And these are all the different plants they have proposed to work. Maybe some of the plants we might be knowing, like azai, that some of the species we may not be knowing. Okay, so once if you are interested, you can go and if it is available in your area, you can go and collect, and you can work it on any of the molecule to target the proposed. So these are all the different reported molecules. They have done so many bioinformatics work. Okay, because why don't we work that and all? So these are all different cancer drugs, actually. Colchicine, Paxlovid. Recently also you would have seen in the news, colchicine is approved for the treatment of your SARS-CoV. So similarly, the aspirin, morphine, and penicillin, syngocides. So these are all now reported as a possible drug for the SARS-CoV. So the current treatment strategies include the repurposing of existing drugs. So we don't have the time now. So we don't know isolate பண்ணி structure identify பண்ணி எல்லாம் வந்து பண்றதுக்கு because we are already burning. So அதனால என்ன பண்றாங்கன்னா that include the repurposing of existing drugs. ஏற்கனவே ஐடென்டிஃபை பண்ண ட்ரக்ஸை வச்சுட்டு அதை எப்படி மாடிஃபை பண்ணி எஃபெக்டிவா வந்து you can use it against all RNA viruses like SARS, MERS and the Ebola. And the remdesivir and RNA-dependent RNA polymerase inhibitor, this exhibited a potent activity. So, நீங்க கிளாஸ்லயே வந்து ஸ்டூடெண்ட்ஸ்க்கு சொல்லி கொடுக்குறப்போ ஒரு ட்ரக்னா அந்த ட்ரக்கோட விட்டுறாதீங்க. So you just explain them the mechanisms. இப்போ கால்சிசின் அப்படின்னா we know it is a spindle fiber inhibitor. Okay, so கேன்சர்ல இருந்து கால்சிசினோட ரோல் பார்த்தீங்கன்னா cell division நடக்கிறப்போ அந்த spindle fiber வந்து it will inhibit. Similarly, you cannot just teach them the mechanism. Then only they will start think of starting any kind of a research in future that will help them. And similarly, the 3 chymotrypsin like proteases and papain like proteases have emerged as the potential therapeutic targets for drug discovery against coronaviruses, owing to their crucial role in viral entry and host invasion. So they feel this will all have an effect on this and prevent the entry of the viruses. Okay. Now we'll enter into the HPLC. Okay. How many of you handle the HPLC? Did anybody has experience? Yes. Yes. Am I audible? Did anybody handle the because I've seen only my slide because I put it in the slideshow. I cannot see your chat and all. You can unmute if possible because I just want to know your depth. Then accordingly I can go. Did anybody have experience, hands-on experience in the HPLC?
Yes, madam.
Thank you, sir. Thank you. Okay. So, one of your friends are having experience in HPLC. So, maybe for rest of them, okay. If you know HPLC, like you just neglect like because I want to start from basic, then we'll see the demo. Okay. So HPLC நிறைய types இருக்கு. So first this is a very essential instrument for the preliminary screening of your drugs. Okay, so you have to preliminary screen your drug. Okay, initially what is the instrument we usually use? Spectrophotometer use பண்ணுவோம். அதுல என்ன பண்ணுவோம்னா like we usually quantify what is the DPPHS இருக்குன்னா antioxidant activity protein இருக்குன்னா we have a protocol laureatal. So laureatal வந்து ஒரு pioneer scientist in science. So still we don't have the alternate for the laureatal, but still we believe it is giving 100% accuracy of quantifying the protein. Okay, similarly we use spectrophotometer where we measure because we have a standardized method, but this is for unknown compound. You may not be knowing, you know only the plant, you collected the plant, you want to screen the plant. So where you will go, only you know how to take it. Even taking an extraction, here in this talk I didn't cover any extraction method for the protein, for the peptides, for the enzymes, and also for these metabolite drugs. So you have a different extraction method. Okay. For you need because HPLC will give only the result. The result is depending upon your sample preparation. So sample preparation is very essential. Okay. So this HPLC is a powerful tool in analysis of any drug. Anything can be analyzed, not only the alkaloid, steroid, அப்படி கிடையாது. You can analyze the peptide, you can analyze the carbohydrate, the sugars, whatever may be, you can analyze. So this is also used in the diagnosis. நம்ம யாராவது டயபெட்டிக்லாம் இருந்தீங்கன்னா லைக் லேப்ல போய் சாம்பிள் கொடுத்தீங்கன்னா நம்மளோட கிளைகாசிலேட்டட் சுகரை வந்து குவான்டிஃபை பண்ணி சொல்லுவாங்க. அந்த ரிசல்ட் எடுத்து பார்த்தீங்கன்னா இட் இஸ் குவான்டிஃபைட் பை யூசிங் HPLCன்னு இருக்கும். So this technique used for separate the mixture of compounds and can be used to identify, quantify the individual. From this all theory we know. So தெரிஞ்சவங்க விட்டுருங்க. Maybe it may look basic, but for தெரியாதவங்களுக்கு, so this is a high performance liquid chromatography. The mixture is separated using the basic principle of column chromatography. So நம்ம ட்ரெடிஷனலா என்ன பண்ணுவோம்னா we use column chromatography. Still we use column chromatography for separating any enzymes or proteins. Okay, so அதே காலம் தான் இங்க வந்து மெஷின்ல வந்து we kept it as a machine, and then instead of the gravity we use here the artificial pressure. Okay, so the sample to be analyzed to introduce in a small volume. Okay, so பாத்தீங்கன்னா 1 ml 2 ml அவ்வளவு சாம்பிள் நீங்க இன்ஜெக்ட் பண்ணீங்கன்னா போதும். Stream of mobile phase. So mobile phase that we have to work it out. நம்மளோட காம்பவுண்ட் போலாரா நான் போலாரான்னு பார்த்துட்டு அதுக்கு தகுந்த மாதிரி we have to design the mobile phases, acetone, nitrile, water or in combinations, அந்த மாதிரி you have to design. And here the sample molecules are retained by the specific chemical and physical interaction with the materials of stationary phase. Here there is nothing but the column. I will show you. Okay, enter the column. ஸோ இது எல்லாமே எதை பேஸ் பண்ணியிருக்குன்னா ரிட்டன்ஷன் டைம் அண்ட் ஐடென்டிட்டி ஆஃப் அ பேராமீட்டர். இது நீங்க எல்லாம் சொல்லி கொடுத்துருப்பீங்க டிஎல்சியில, பேப்பர் குரோமட்டோகிராஃபில, உங்க ஸ்டூடெண்ட்ஸ்க்கு எல்லாம் நீங்க எடுத்திருப்பீங்க. வாட் இஸ் த ரிட்டன்ஷன் டைம்? அது நான் உங்களுக்கு என்னன்னு சொல்ல போறது இல்லை, பிகாஸ் யூ ஆல் நோ பெட்டர் தென் மீ. ஸோ இயர் இந்த ரிட்டன்ஷன் டைம் டிபெண்ட் அப்பான் யுவர் சாம்பிள், composition of your stationary phase, and the mobile phase, and temperature, and இந்த எல்லா பேராமீட்டர்ஸும் தான் நம்ம வந்து நம்ம பிரெயின் யூஸ் பண்ணி ஒர்க் அவுட் பண்ணணும். ஓகேங்களா? ஸோ the time taken by the sample to travel from the point of injection to the end of column. ஸோ நம்ம அந்த காலத்துல டிஎல்சி பேப்பர் அந்த காலத்துல இல்லை, இப்பயும் பண்றோம். ப்ரிலிமினரி ஸ்கிரீனிங் பண்றப்ப என்ன பண்ணுவோம்னா ஸ்கேல் வச்சு மெஷர் பண்ணுவோம், எவ்வளவு வந்து சொலிட் மூவ் ஆயிருக்கு அண்ட் சால்வன்ட் மூவ் ஆயிருக்கு, அதை வச்சு டிவைட் பண்ணிட்டு வில் கால்குலேட் த ரிட்டர்ன் டைம். ஹியர் you have to analyze. Yes.
Kindly speak in English. We are not able to get it. I'm so sorry.
Okay, sure. In between, I use only few words. I will reduce it. Okay. So here, you use and because I didn't ask Prema that who are all the participants. I thought that mostly from here. So I used for the comfortability. No problem. I will reduce in Tamil. So here. This is the thank you for expressing. Okay, that means you are listening to my talk, because I'm worrying about whether you people are listening or not. Okay, so this is the retention time. You might have calculated for using a paper chromatography or the thin layer chromatography. So same here, you are analyzing to in the HPLC, but here only instrument will do. There we manually think. So this is the workflow setup. Anyway, I'm going to show the real video, and so this is the components of the HPLC. So I told you about the mobile phase. So in the mobile phase, we call it as the reservoir. This you have to standardize for your mostly the acetone, nitrile or methanols are used. Okay, so methanol is a universal solvent. Why? Because we use most of the molecule will dissolve in the methanol, so we use the methanol as a solvent. So most of the molecule will dissolve. Here we use the mobile phases, and you have a pump. So this pump is the binary pump. Here you have a 2 pump. It depends upon your application. Either you can use a single pump or the 2 pump, and you have a sampler, and this is your stationary phase or the column. Okay, this is your stationary phase or the column. So you could see here and I'll show you the real columns, and you use a detector. A detector also you have to choose depending upon your sample. Suppose if you are giving a sample for outsourcing, you have to ask what detector you have, whether you have a UV detector or whether you have any fluorescent detector, or if you have any PDA detector like that, you have to ask, and then you have to give the sample. Okay, then you have a software and things. So you will be, this will be the major pump, and you have a mobile phase reservoir, you have a pump control module, temperature control module, and in my lab I have PDA detector. I don't have a UV detector because PDA will also serve as a UV detector, and you have a Empower software. So this is an improved form of a column chromatography. I already told you. And the difference here, instead of dripping the solvent under gravity, okay. So what that is what we do in an thin layer chromatography and the paper chromatography. Here we maintain a pressure up to 400 atmospheric pressure is applied on the chromatography, and HPLC can what I have a system which can go up to 6,000 psi. And a very smaller particle size of column packing material is used. And in solvent reservoir, the solvent reservoir store the solvent or mobile phase to supply the column as necessary. The solvent is pumped to the column in a specific flow rate. This parameter you have to fix it. I will tell you how to fix it. And you have to inject the sample. After placing the sample in the sample reservoir, in the injection process fully automated here in HPLC. The injector injects the sample into the continuously flowing mobile phase stream and carry the sample to the HPLC column. Then finally comes the detector. So I told you different kinds of a detector. Here one of the method is the UV light. Many of the compounds absorbed by the UV light of various wavelengths. Here the UV is signed when the component passed out from the room. This light is passed through the component and absorbed by it. On the other hand, there is a detector to identify what is missing in the UV lights. The amount of UV observed depends on the amount of compound passing out the column. So these are all some theoretical steps. So this is the detector which I have in my lab. This is a photodiode detector, which is similar to UV detector. The range will it can detect up to 190 to 600 and more than 700, 800 nanometers. And the resulting spectra is three-dimensional plot of responses wavelength. Advantage, the wavelength need not be selected, but detector response of all common. So for this will be useful for unknown compound. Okay. So if you know the known absorption of nanometer, you can fix it up. For the unknown, you have to go for screening, right? So you can screen. You can send the sample. You can detect at what wavelength your compound is getting a peak. Similarly, you can choose either one pump or the two pump. You can mix up based on the polarity. You can mix up the solvent. You can go ahead. And then you have a pump control where you can use a maximum up to 6,000 psi. The stationary phase column, it will vary. Accordingly, you can choose either C8 column or C18 column. And the particle of the stationary phase is greater is the resistance of the flow of the solvent. Hence, the pressure is the recommended here. So we have a column temperature controller for obtaining a better and reproducible chromatogram. Temperature is very essential, and this also you have to standardize. So now we'll go to see here. Just check whether this video is okay, or I will go for a other way. Direct video, please. So this is my department because I shot it in the mobile. It would be larger if I used the camera. So this is my department. I told you, and this is my lab. Is it visible? Video is visible?
Yes, ma'am.
Yes, madam.
Yes. Thank you. So I told you we have a different discipline of lab. So in presentation, I cannot share a very big video file. So I have taken a small file, and this is my lab since 2003. There was no lab there, and after getting the fund from the DRDO and the other DSP, DPT, and many other funds, so we have a complete facility for doing a plant molecular biology because I'm expert in the transgenic plants. Mass spectrometry is my passion. Okay, HPLC and mass spectrometry is my passion. But the main hardcore theme of my lab is production of transgenic, especially on metabolic engineering, to increase any of the pharmaceutical and the nutraceutical industrially important compounds. So this is my lab actually. If they would have arranged the talk on the daytime, I might be presenting. I was having intention to present all these from my lab. So instead, just 2 days back, I took all the videos. So we have a complete setup to do the plant molecular biology work. So okay. So now we have a separate facility for this is my lab, and we have a different separate facility for HPLC and flow cytometer and high-end equipment. We have a separate facility for the high-end equipment. It was so breezy in Coimbatore. Now the climate is so rainy and so breezy, and this is a funded. So hope you colleges also will get the SAP and FIST fund. So under that fund, we purchased so many higher end equipment where this room is completely having the workflow for the flow cytometer, and this is the cryotome. Cryotome, you can use it for taking any section. Because we purchased one high-end fluorescent microscopy of about 25 lakhs. So for that, this is the ChemDoc where you can visualize the DNA and proteins. Okay, so this is my HPLC facility. So this instrument, like my kit, I'm maintaining this for last 10 years. 2008, I purchased from the Waters, and so many PhD, so many samples. I usually do as a free of cost. We never charge for any people, and so many plants has been analyzed, samples has been analyzed by using this. So coming to the components of the HPLC, you need to have a software, right? So I have a Empower software, and so because everything is controlled by this, if any in between, if you want to stop also, I will stop the video. Just tell me. And I told you this is the binary system. So this is the two pump. You could see upper there is a pump and down there is a pump. And some of the sample you will be using a two pump. Okay, so two pump system. And here you can keep. So this is called we are purging the sample. Purging means like you can run out the residues. So this is how you keep your mobile phase. Okay, so this is we call this mobile phase reservoir. So all your syringe filter we use for filtering out our samples. Okay, so this is the control model. So this is a different. So you can see the column. This is the injector port, and this is the temperature control model where you can control your temperature. This is the injection port where you have to insert your sample. Okay. So this is insert. This is the compartment for column where you keep your different column. Okay, different column. You can just open it, and the screw you can adjust it if you want it. So below is the detector. Below is the detector. Okay, this is the PDA detector. That is a photodiode array detector. I have this. This is the complete setup. So it is written right photo array detector. Okay, so it has the wavelength up to directly 750 to 800. So after the analytes will be collected. So chromatogram will be recorded. The remaining solvent and sample will be collected. Even sometimes we'll reuse it. because we are all Indian universities now, sometimes we will use it. And also, this is along with that, we have complete setup for how sample preparations and other facilities are also available, which I'm not able to cover everything. Um, this is the complete setup, and you will be having different columns. Okay, different columns. This is the injector needle used to inject your sample. You will be injecting only or 1 ml or 2 ml of sample. So you should be ready with your sample. Suppose if you want to work it with the HPLC. So this is whatever I shown in that previous flow chart, that complete setup. So I could see, you could see some columns, symmetrical columns. This is the boxes which has the column. Okay. Now we'll enter into the sample analysis. Okay. So can anyone able to identify the sample? Okay. The sample, this is one of my DST project. The sample is Senna alexandrina. We call it the Spinelveli Senna. This is the potential plant where we export this leaf to the abroad for taking a laxative drug sanicide. Okay, so we usually standardize the method how we can isolate the sanicide. Maybe here some of the methods are cut shorted. Okay, so we are not able to take full protocol. Like once if you want to analyze the sample, you take out the sample either from the in vitro and in vivo, and use the liquid nitrogen or methanol, any whichever based upon your compound, you can use any solvent to take. And all the sample has to be filtered, because otherwise your column will damage. All the sample has to be filtered. We use here the 2 micron syringe filters, and see the samples how it is getting filtered. Like so, take the sample, grinded sample, and here we before that we will prepare the polar and non-polar separation. So many steps are available. Okay, and then it is a partially purified compound. Either you can use for a oxalate or maceration method, and then after that, before injecting into our HPLC, the sample need to be filtered. This is the 2 micron of filters where all your dust particles, impurities will go. So you have to standardize every. But if you use a oxalate, it will give a pure extract. It will go, or maceration or sonication. Any method you can use it. Okay. So then you have to prepare the mobile phase. So mobile phase, either maybe of acetonitrile water, anything. This is the sonicator, water bath sonicator. So sonicator you have two types, probe sonicator, that is used for tissue. Here you have to sonicate your mobile phases for the complete dispersion of your mobile phase. Also may contain some of the small particles. So you must be careful when you are using a sonicator, because it will create a very disturbance. Sometimes if you work continuously, your ears will get damaged. So people will keep the sonication for half an hour, they will come. And then you transfer the mobile phase to the HPLC. So if you want to use a single pump, you can use a single pump. Sometimes in ratio you can fix it up. Pump for A and 2 pump you can fix it up. So you are just now you are transferring our HPLC mobile phases to a newly prepared mobile phase. So intention here to analyze the sanicide. This is a laxative drug. So we use the acetonitrile methanol. Now I am going to use only a single pump, isocratic pump, where I've given the run sample. Now this software. This is very essential. So injecting the sample, preparing, it's all easy. You should know about that too. Borders is having a power software, and you have to get trained in the software. You have to set the program and other parameters has to be set before injecting your sample. Now the sample is ready, system is ready. You go to the software, you set up your program. What are the parameters? That's what in theoretically I told, like what are all the parameters: temperature, pressure, flow rate, nanometer, everything you have to set. So once if you make a method, again and again you need not standardize. Once you can save that program as a method. When you run next time a sample, you can choose that method and apply, and you can run the sample. You can save it in your name or any. So the main parameters you have to choose, like your flow rate. You have to give how much flow rate, and what is the if it is a non-drug, you can give the nanometers. What is the nanometer you want to analyze? If it is a toxicity. Uh, so you have to set the pressure, and then flow rate is the injection volume. Hope it is self-explanatory. You can. If it is unknown, means you have to give a range. You have to give a range of nanometer, like 190 to 500. You can scan the chromatogram. So how many peaks are coming, and then you can in HPLC you have a preparative also. You can isolate those compound. If you have an automatic sampler, automatic liquid collector, you can collect those samples and you can do your animal experiments, like activity you can check, either antiviral activity or anti-cancer activity. So now you can see now the baseline you have to set. So first you have to set a baseline. You have to check whether the instrument is in a proper. If any contamination in the column, then you will not get a baseline. Means it is like a standard of HPLC. Okay, and sometimes if it is a known compound, you can use some standard molecule also. Okay. So now we can see the chromogram is start coming. The baseline, we are setting up the baseline to check the instrument. Sometimes if it is you are very confident that you are previously also running the same sample, like if you are very confident, you can stop within 1 or 2 minutes and you can start analyze your samples. If you are a newcomer, like you have to go and set the things. Okay. There is one video is Miss Anchi. So this we have seen. This also we have seen. Only the sample introduction video. That's just minute or only 1 minute I will show that. Whether my slide is visible?
Yes, madam.
Yes, ma'am. Yes, ma'am.
1 minute. That is one video. Let's watch. So this video, you can see how the sample is getting injected. Okay. So now the vial, hope you remember the vial, which we already prepared the samples. This is the smallest needle which we use. So I've shown already the injector port, right? I've shown already the injector port. Now the program is already we standardized. The baseline also standardized. We confirmed that everything is pakka, and we set up the program. We set up the nanometers and flow rate, temperature, pressure, everything. We set up. It will take. It is really a time-consuming process. Because usually from the people they will give the sample to the outsourcing. They will ask for the result. But working with this need a very patience. If you come back after, so this is the sample injector port. You can see, you can inject the sample. So now load the sample. Now the sample will be running through your column. The sample will be running through your column. Then after injecting, now we could see from here it will start developing a chromatograph. So if it is a non-filtered or contaminated sample, you will get a clumsy of peaks. If it is a pure compound, you will get only a single peak or double peak. Or if you want to analyze profile, if you want to do a profiling, so what you can do, like profiling, you will get many peaks. What you have to do, whenever every peak comes, you have to collect the aliquots. That aliquot you have to work it for the biological samples, biological analysis like cell line cultures, whatever activity you want to check, antivirals or whatever one, you can use it. Okay, so then it will start developing. Chromatogram will start coming in your system. Depending upon your sample concentration, how much concentration is there, like that. So if you take a crude or if you take a bit of lip sample, you will get nothing. You have to take a kg of samples and take a concentrate, do any oxalate or any extraction method and concentrate using rotary evaporator. If it is a protein, you use the column gel chromatography and you purify the protein, concentrate the protein, and you dissolve the samples, then you inject it. Okay. So if your sample itself it is very mean, then you may not get a chromatogram. It depending upon, even I used to analyze some of the recombinant proteins, some of the recombinant. See, nice peaks, nice many peaks in this. In 3 to 4, the retention time is our rest of compounds. This is our Dalcolox compound. Okay, so after injecting, you can take out the needles, and by using, you can save the chromatogram, and now you can use it for your research purpose. Okay. So a fine chromatogram will look like this. A fine chromatogram. So this is a andrographite. So we analyze this paper we have published, like it's not a very good journal, but we published like so this having an andrographite from Andrographis paniculata, because nowadays people talking about this plant for analyzing the undergraph lead content undergraph plant, because this is used in your Kabasara Kudinira or something like that. So this good chromatograph will look like this. Okay. So I told you we work on the transgenic plants, and this was our first defense DRDO project where we transferred a gene from the Arabidopsis to the tomato for conversion of gamma tocopherol into the alpha tocopherol. So this is entirely a different kind of a research, and this is all the transgenic process. So after transgenic, see the nice chromatograph. This was appreciated by many of the scientists. So you can get the separation of the gamma tocopherol, alpha tocopherol, and you can find a very nice chromatograph like this. So if you are having a very chromatograph, it is easy to publish in any good journal. Okay. Now we'll move on to the mass spectrometer. So I'm done with the HPLC. So mass spectrometry, the direct mass spectrometry video I'm not able to take. So what I've done, I prepared some workflow which already have standardized. Okay. So we have seen the HPLC, like you can analyze based upon the chromatogram, you can confirm the drug. But what is the drawback in this? Why do you want to go for a mass spectrometer? Okay, so some of the compounds will have a same retention time, and this even if you use the same mobile phase or same pressure, same temperatures, but there will be a minute difference among the two isoforms. Okay, so in such cases, you have to confirm the structure of the molecule. There you need a high refined instrumentation method, that is a mass spectrometry. Okay, so you have a different types. You have so you can analyze any of the molecule using the mass spectrometry. So in mass spectrometry, it is not a single instrument. Okay, it is like an ocean. You have a different mass spectrometry. You have a gas chromatographic mass spectrometry. You have a liquid chromatography mass spectrometry, and you have a MALDI-based mass spectrometry, and it has a different mass spectrometry. This was my first training in 2011 at the South Korea. This was the lab at Tongki University, Suwon. They had a very nice facility, and there only I started learning my mass spectrometry. Since then, I developed my interest on the mass spectrometry, and after 10 years, now only I am going to achieve purchasing all those instruments in our university. So this photograph was taken in 2011 at the South Korea, where I trained on the mass spectrometry. So this is the plant which is present on the Rhodiola rosea. It's a high source of cancer. So similar to the HPLC, this also has a workflow. So here you can choose the mass spectrometry, whether it is a gas chromatography based or liquid chromatography based on your compound. Okay. So if I use any volatile compounds or any oil compounds which can withstand a high temperature, I can go for a gas mass chromatography. Okay. Or if my compound of the drug is very sensitive to the temperature, I have to depend upon the liquid mass chromatography. If we are using a protein, you have to choose the MALDI, and other things are detectors are will be common. So what is the principle in this? Okay. So we have seen in HPLC the principle, and based upon the principle, how you are using it. In mass spectrometry, what we are doing? So this is your analyte. Analyte means your drug molecule or the moieties which contain your chemical entities. So you have to ionize this compound because you have to know the molecular mass of the compound. So how do you measure the molecular mass? Here the analytes are ionized by using a different ionization, and these ions are deflected in the magnetic field and then detected by using a mass spectrometry. Okay. So there are two different ionization. We have electron ionization and chemical ionization. Electron ionization uses the heated element to produce the electron. Chemical ionization uses the reagent gas methane to produce the electrons. Okay. So this is based upon the principle every molecule has its own molecular weight. Based upon that, you can identify the compound. Okay, so this is a very sensitive tool. You can go further defining your structure, molecular weight. So you can confirmly say this is my drug, this is my compound, like that, because it has the sensitivity to detect the 5 ppm or 0.005 percentage of the compound. If it is present even in this concentration, the instrument can detect. And this is the way to charge an atom on the molecule. The basic principle here, you are charging your atom of your drug molecule, and you have to place the charged atom or molecule in a magnetic field or subjected to an electric field and measure its speed or the radius curvature related to the mass to charge ratio. And they detect the ions using the microchannel plate or the electron multiplier tube. So this is the principle. Okay, here also LC will be there. HPLC will be the front end. Mass will be attached to the HPLC. Okay, instead of PDA detector, you will have a mass detector, and in between you will have your ionizations and the quarter pole. Everything will be present. Okay. So it is like an ocean. We cannot cover in 1 hour or 2 hours because each mass spectrometry may require 2 hours of presentation if you completely talk about. So where your sample is getting ionized, then it is subjected to the mass analyzer. It is detected by the detector. Okay. Here, the ion after ionizing the compounds are transported in between the magnet. Okay, so only here to prevent the scattering of your molecule, to make the path to send to the detector. Okay, so here similar way this is getting recorded. This is the instrument setup. You have a triple quarter hole here. You can mask and collision cell and MS2. So here you can get the two. So MS, you would have heard about GC, LC, MS, LC, MS, a triple quarter hole. There are different technologies there. So here this is a LC, MS, MS. So what is the ion source you will choose based upon your molecule? Whether you want to go for a soft ionization or the hard ionizations, based upon that you can choose the ionization method. So this is the traditionally used ionization spray in all other research. This has been adapted for ionizing molecule in the mass spectrometry. Here this is a schematic diagram where you could see the sample is injected and is getting ionized by using ionization. Here you could see the sample code and how this is going inside and how this is the quarter poles. So how the quarter poles. So in this you will have a different model, split and splitless model. When you give a sample for your analysis or outsourcing, they will ask the programming. Most of us, we may not be knowing what to give as the parameters. So we simply give the sample, they will analyze the sample, and even some of them, they won't even give what is the protocol they followed, and they will give the result. We'll keep it in our thesis. That's how story will finish. It is not like that. For your compound, you have to tell what is the nature of your compound, whether you want to run it in a split mode or splitless mode, what ionization you need, whether it is a soft, if your compound is very sensitive, then soft ionization you need, and if it is hard, you need a hard ionization. And you have to, if it is a GCMS, you have to derivatize the sample and then you have to give it for the analysis. So many pre-work you have to do. Okay. So here you can see how the ionization source is present. So here this is the ion spraying needle. Here sample cone where you can keep your sample, and you have a vacuum valves. So similarly, your sample is getting bombarded. So when you do an electrospray ionization, so this is the theoretical part. and your sample inlet nozzle, so it will get a charged droplet of your molecule will be separated. Here it is atmospheric pressure chemical ionization source. Here the sample is getting vaporized, and here you could see again the corona term, because the corona is the common terminology for this kind of structures. Corona mimic the same structure, they also put the name. So here you have the analyte, and it is splitted by the splitting cells, and then it is getting electronized or ionized. So here you can use the MALDI term. This is only for the protein. This is only for the, here you can see the IgG, the spectrum of your IgG. For the MALDI, the front end should be your 2D gel electrophoresis. Okay, you have to run the sample in 2D gel electrophoresis. Then from that you have to pick up your protein. Then you have to give it on the MALDI toughness. This is the mass spectrum of your progesterone. Why I am giving all this example? All this wide variety of sample can be analyzed by using a mass spectrometer. So this is the typical mass spectrum where you can have your abundance of your isotopes. Okay. So here you can see still further how the sample is going inside the inlet, and how the filaments are spraying the electrons on the samples. The neutral molecules are filtered, only the charged molecules are transported. Here you can see, for example, how the ionization change the fragmentation of the molecule. Every fragment will produce its isotopes of your spectrum. So this is, we generally call it as the quadrupole mass filter. This consists of your 4 parallel metal rods with the different charges. The applied voltage affects the trajectory of ions, and the given DC and AC voltage, only ions of a certain mass to charge ratio pass through the quadrupole filter, and all other ions are thrown out of the original path. So the detectors produce either it may be a ion channel, an electron multiplier produces electronic signal via the secondary electronic emission. The timing mechanism integrates these signals with the scanning voltage, and the instrument report the M by Z as a struct of the detector and need a constant and regular calibration. So you have to standardize. This is a schematic diagram for the GC-MS. This is the GC column, same as that of your GC chromatography. You have a GC column O1, where the column will be different. Here the column will be, you will have a circular column, and here you will inject the sample, and the ion source is present, getting analyzed using a quadrupole, and then data are recorded. So the advantage of mass spectrometry here, you can use it for a profiling, complete profiling. Nowadays, even in animal human sample, they do complete. Even in the COVID, they have done complete analysis, like metabolism analysis. You take out the sample, you give it to a mass spectrometry, you can completely analyze what are all the metabolic changes happen. That kind of research can be done. Anyway, you need an advanced model of your NMR FTR for identifying an unknown compound. Libraries are there. By using the library, you can identify the compound. If it is unknown, you have to go for a FTIR or NMIR for the structural confirmation. And this is useful for identifying the suitable source of plant for industrial production. This was my first mass chromatograph. Sorry. So here you can find that this is the metabolic profile. This is because I'm a plant scientist. I work on the herbal plants and the food crops for the industrial purpose. So this I took this sample to South Korea and I analyzed it. Okay. So using the mass spectrometry in the Korea University, South Korea, this was my first chromatogram. So I was excited to see what is it. Even she doesn't know how to explain it. And then after that, day by day, after that analyzing them, I came to get an experience on how to analyze the peaks and how to count the ions, daughter ions, and to confirm the structure. So it is also can be used in the human research, like how we can choose the human and rat serum sample, and you can use the blood proteome, and you can digest, run the sample, and take the after trypsin dissociation, and you take up the sample, and you can use the solid phase. Many extraction methods are nowadays available. Even solvent free extraction methods has come. Now you need not use any solvent. You can use the gases for extracting the. Now I'm buying such kind of instrument in our university with the thermal desorption, where you can analyze only the volatiles. Take out the sample, any breath sample you can analyze. Okay, so you go and take a breath sample of any patient. Any patient, you collect the breath sample. You need not go for a blood or urine sample. Just suck your air, let them blow, and collect the sample in a canister bag. You just give it to a thermal desorption, which is combined with the mass spectrometry, and you can analyze what are all the profile, what are all the profiles. So in future will come, like you need not go for a RT-PCR test or any other blood test. Only from your blood sample you can diagnose the diseases. So nowadays it is possible. It is at our door. So next year we'll have this instrument in our department. And you have data. Only you should have a knowledge and data processing, how your data and softwares you should have a sound knowledge. So I just want to tell about my practical thing I have done for my sample, how I prepared the sample and how I obtained my photography. So I collected the sample from the field grown and in vitro cultures. I separated into the polar and non-polar phases. This was the methodology I standardized. You never get this methodology in any textbook or any Google content. Okay, so I tried with a liquid nitrogen sample for grinding. All these photographs were taken in South Korea. Okay, then I separated, I agitated, and then I added, I separated, I microcentrifuged, and then I separated the polar and the non-polar sample. I used here methanol as a solvent to prepare this. I centrifuged the 10x of the sedimentation value, and then this was the sample I concentrated. Like after that, because nowadays the students will get lazy in preparing the sample. They will take out the leaf or any animal sample and blood sample, they will give it for the analysis, they will demand the result. It is not like that. Result is on your hand. How you prepare the sample, that is very essential. So you just train the student that how to prepare the sample, that is very essential. Okay. So you have to separate out this. So you could see here that there are two phases of the samples, and by using the rotary evaporator, you can concentrate. So this much sample has come to only this much of residues. Okay. Similarly, you have to try another sample. Then only you can pull out the content before injecting into the mass spectrometer. Okay. Here I used this is derivatized agent. Okay. So for if you want to do a GCMS, you have to derivatize the sample. And then I obtained this sample preparation for LCMS. I standardized the method for the LCMS, like liquid nitrogen, same method. Here I used the sonicator for because that is for a GCMS. There I used a hot plate. Here for the LCMS, I used the sonicator, and then I filter out. Then I did my LCMS sample injection. So this was my ion chromatogram. So whenever you are getting a chromatogram, you may not know what is it, whether it is ion chromatogram, and this is a ion chromatogram. From that, you have to get a mass spec. This is the see you are exposing your compound to the ionization. So each isotopes of molecule, you will get the abundance, ion abundance. So totally you will calculate. Then this is here the compound detected. Although you see so many things. Here the abundance is a 204. Okay, what is the compound? You have to identify. Here also 204. So all the things I am getting 204 compound. Okay. So this is the ion chromatogram of a LCMS. So you have to, so I run it in positive mode. Okay, positive mode. This is again spectra ESI scan. So this was the statistical analysis. What is the wonder in this graph? If you see, like you don't know what is there in your sample. Okay, you don't know what is there. What is that? So you can use this software. You can group the sample. See, this much of sugar alcohol is present. This much of nitrogenous compound is present. This much of simple sugars are present. This much of compound sugars are present. Okay, so you can group it out. You can group the sample. So you have to play with this. Is a pie plot opium. Some people will be doing this in a biodiversity study. Same methodology used here to group the compounds. Okay, you can identify a different samples. What is the diversity? Whether it is having a sugar, alcohol, high or low, it's present or not. You can go for a specific target. So this was first time I identified the thymol as a new compound, and rest of the things. These are all the molecules which I have identified from my mass spectrometer. So this is the plot again. Each color represents the color. So among the samples, I confirmed that the metabolic. Even I detected it was. I could able to find there are 1,500 metabolites were detected. Just imagine. Okay, that is the beauty of your mass spectrometer. Okay. And first time I detected the thymol in the culture. There are 600 metabolites in the in vitro sample. Hence, I standardized this method for the metabolic profiling. It is suitable for the industry. So now you identify the drug either for the cancer, either for the corona, whatever the disease which you want to work. You identify the drug. So what is the next step you want to do? Like you have to go for the structural elucidation. So the mass spectrometry has different libraries. For specifically, if you use gas chromatography mass spectrometry, and it has a different library. If you have a known compound, it will hit the library. It will tell you this compound is present. If it is not there, what you will do? Okay, either you can develop, or what you can isolate that aliquots. I'll show you. You can isolate the aliquots. You give it for the FTIR or NMR. Now there is a supra instrument has come, LC-NMR. Maybe it may cost about in many crores. Like if you simply run the sample, this will analyze your sample. It will both in LC and it will subject it to the NMR. It will give the complete structure. Maybe in future, in 1 or 2, 3 years, some of the institute will buy this instrument. So everything is based upon the instrument only. But what is our responsibility? Like the biological activity, we have to confirm. For that, either you depend on saline or in vitro assays or the animal experiment. If it is antiviral, you should have a biosafety level 3 lab. Unfortunately, only few institute like here in, I think in SRM and other institute. Am I audible?
Yes, madam.
Yes, thank you.
Yes, ma'am.
Yes, ma'am.
Yes, okay. So you have few institute which has the biosafety level 3 lab, where you can tie up, you can give your compounds and you can work it for the antiviral property. Okay. So what is my opinion? Elucidation of drug, it's not a big task. Only thing you have to confirm the activity of the drug. Okay. So and also you have to identify whether the compound which you identify is the druggable compound. What is the difference between the drug and the druggable? I told you the ADME parameters. You would have studied absorption, desorption. So how it is absorbed, dissolved, metabolized and excreted. How long it is taken inside our body. And also the solubility. If you have any friends with the pharmaceutical research, the D-Pharm, M-Pharm, D-Pharm, the pharmaceutical college, you just tie up with them. They will be having, because here I know one, it's in Naipur, it is in Chandigarh, where I used to work with them. They have a complete setup for the pharmaceutical analysis, drug analysis parameters. So we may be wondering for simple things. You have solubility. They have a simple experiment. They take the test tube and they will insert the compound or the drug compound. They will study the solubility and the injection how it can go. And also you have one more instrument called the photon detectors, live cell imaging photon detectors. These are all essential to identify your drug properties. Okay, so I worked with the photon imager where you can anesthetize the animal model. Rat you can anesthetize. In tail you can inject your drug molecule along with the marker dye, and in 5 hours, 4 hours you put that in chamber, and it is connected with the imaging softwares. You can imagine how this drug is going and targeting inside, whether it is going to the membrane or which organ it is going, whether it is going to the lungs or the heart or the brain. Like that, there are facilities are there. Then you have to collaborate with them, just work with them to identify the properties of your drug. So similar experiment you can do, or for the simplest, every lab in our department they have animal cell line and animal house. Or they use a different working models like zebra fish. So many models are there, like okay, and also some worms are used. Similarly, you can choose any model because the getting the approval for the animal experiment, you need some committees, ethical committees, and all this all will hurdle for us, for the especially for the people who do the drug research. These are all hurdle for us. And those who are expert in animal experiment may not be knowing a complete detail on the drug molecule. So you have to collaborate. That is the only way uh, to uh, to get a new drug, successful drug. Okay, so you have to analyze your drug. We evaluate through animal experiment or the cell line experiment, and the drugability has to be analyzed. Then the toxicity, even if you identify, then the toxicity problem will comes. Okay, so what are the how much dosage you have to fix it, and also you have to prove it doesn't affect anything, and so many toxicity assays, so many assays they will do, like a Comet assay, so many assays they have for the cytotoxicity and toxicity. And also nowadays you can use the flow cytometer for the chromosomal aberrations. So everything, after analyzing all these parameters, you have to file for a patent and the approval. Now the ICMR is giving importance. If you come across with the other results of the experiment, now the 2G glucose drug developed by INMAS has given the permission for the COVID. That was my first funding agency. I got my first project from the DRDO INMAS only, and the OSIM, and there was a project called Rosmanic acid. In such cases, what we'll do, even a soft people like me will be cheated by many big institutions. Like once if they identify that you have a potential target, first project they will give, second project they won't give, but they will work themselves to get approval. So such kind of hurdles will come. So don't bother about that. Whatever is there, it will come for us. Okay. So similarly, you can tie up with the institute. And analyze your drug. You can file for patent. That is very essential because it will protect your technology, and you apply along with the ICMR institute. You apply for the approval. So hope you will come up with a new drug for any disease in the future. Okay. So I thank my collaborators, because without them, I would not be an expertise in mass spectrometry, because in and around Coimbatore, if anybody buys mass spectrometry, I'll be a technical expert committee. And this I learned this technique here, this under the ENSA Korea. You can also apply. I think it is you have to go within the 40 years. I went in 2010 and 11. So there are different schemes for senior professor. The INSA is a very common one. maybe after the COVID is getting over. No, the coronavirus is only a 2 ORF. It's very small. Only thing the people are afraid of the toxicity. We have many lead compounds. We have plenty of drugs. Only thing we bother about the toxicity. Okay, so if this is done, it will be easy because virus we continuously come across the problem with the viruses. Okay, even for the normal cold virus, that is also a coronavirus. We don't have the drug. Only we have remedies. Okay, and also we felt it is not toxic, so we just left it. We mainly world millions of rupees have been put for the cancer research. Everybody, if you see anybody, they'll say, I'm working on this cancer, that cancer, this cancer. They forget about the infectious diseases. Okay. So infectious, we have to take much attention on the infectious diseases. We have to work towards the infectious diseases. Unfortunately, I'm not an animal biotechnologist. I'm a plant biotechnologist. I know only if you give any sample, I can analyze and give the sample. What is the sample? Whether it is a pure sample, whether it is a pure drug, what it could be the structure, what could be the thing. Only thing again, because basically I'm a plant molecular biologist. I work on the recombinant protein and the recombinant product. But if you have a facility to work on this animal experiment and you tie up with them, and if you have any new plant or molecules, or if you want to develop any protein or peptide, synthesis of peptide and all, it is very easy. It is like a simple chemical reactions. And also you can integrate with the bioinformatics tool where you can identify the lead. Now bioinformatics. So, but the problem in bioinformatics, whatever we do, 80%, only 20% of the result can be proved in the wet lab. Okay, so whatever you do in simulation, same cannot be replicated in the wet lab. So only 20% will be successful. But luckily, if our molecule is in a 20%, then you will be the Nobel laureate or the Nobel Prize winner. So don't think that you are in a small institute. Yeah, we don't have. Even I was having a feeling like that. Yeah, I'm in an Indian university. I have only a restricted group. Even though I have full-fledged facility for my plant molecular biology work. But now I wonder, even with a higher facility, they are not able to solve the problem. Maybe in future, it will be solved. So we have to concentrate on the infectious diseases. Okay, not only on the corona. We have many infectious diseases. So if that come, that epidemic cannot be controlled. So we have to work towards that. That is my work. So I thank my collaborators. So many of them have contributed my upliftment of my technology. And thank you. And thank you once again. And if you have any queries for this,
for your excellent presentation. Dear participants, if you have any questions, chat box.
Or you can ask me in person.
Yes, ma'am. Ma'am, one question. What detector will be best for plant samples?
No, what molecule you want to analyze? That is very important. Okay. So you have a UV detector, you have a PDA detector, you have a fluorescent detector. Suppose if we want to isolate metabolites, like we can use the PDA detector. Or if we want any hormones, like some people will come, I want to analyze the carbohydrates, monosaccharide, disaccharides. Like that, UV will be best. So based upon your absorption, what is the maximum absorption of your compound? Based on that, you can select your detector.
Okay, ma'am. Next question. Which solvent system is best for the isolation of anthocyanin using HPLC? Which solvent system?
Obviously, methanol. Anthocyanin, all your pigment, methanol is a good solvent. And also, we use aceton nitrate sometimes, but methanol is giving a best result.
Okay, ma'am. Thank you, ma'am. This question from Joseph ma'am, is there any special device for injecting the sample in HPLC? Any special device?
Yes, now whatever I have is a very basic model. Okay. Now the automatic sampler. Now we are purchasing one instrument that will do everything. Only you have to give the sample. That will take the sample, you program it. That will go and insert the sample inside the instrument. So you have an auto sampler. You have the only thing you need money. You can buy all those things. So battery systems are available.
Okay, ma'am. Next question, which criteria should we prefer while selecting between LCMS and GCMS?
Pardon? Can you repeat the question?
Which criteria should we prefer while selecting between LCMS and GCMS?
Yes, that's a fine question. Like, what is the compound you are working? Suppose if you are working on a protein or peptide or any drug molecule which has a heat liability. So if you heat it, that molecule will degrade, right? So in such a cases, you have to only go for a LCMS. Or if you want any fatty molecules, fatty acid profiling, or any molecule which can withstand that, because in GCMS the temperature is up to 800 degree, and we generally fix the temperature in 280 degree, 300 degree. Just imagine your sample will be exposed to in such a high temperature. If I keep any carbohydrate sample or protein sample in GCMS, it will get degraded. Okay. So based upon the compound, you have to choose the GCMS or the LCMS. Temperature sensitive.
Okay, ma'am. Next question. What happens?
Excuse me, Prema. Continuation of that question, ma'am.
Yes.
So can lyophilized sample be used, ma'am? Then.
Yes, definitely you can use it. You can use it.
Thank you.
Another question. What happens if the sample is stronger than the mobile phase in HPLC?
Sample is stronger than the mobile. Yeah.
Mobile
Yes.
phase.
There are. Yes. That also will happen. Even sometimes we feel methanol is having a low polarity than the sum of the compounds. For such cases, you have to go for any GC apparatus, where your GC will have you will be subjecting this to high temperature, or the volatile is your compound. Thereby, you can separate the compound. And also now we have I told you, solvent-free extraction, superfluidic method, carbon monoxide method are available now. That is the strongest method, where you need not use any solvent. That will dissolve. Even if it is a hot sample, that superfluid, critical fluid method, that the instrument will cost about some 50-60 lakhs for sample preparation. You can buy it. And you have a SPME method. Many methods now has come. You can choose. Yes.
Okay, ma'am. Thank you, ma'am. Next question. What is meant by ADME parameters? ADME parameters.
That is very basic in pharmaceutical. The absorption, desorption, metabolism, and excretion. Maybe teacher who teach pharmaceutical biotechnology, when you are starting with your drug molecule, this ADME character, that is the property of the drug. If you consume a drug, how it will be absorbed, how long it will take to get absorbed in our body, and how it is metabolized, how long after 3 hours or if you take paracetamol, doctor will prescribe morning one paracetamol, evening one paracetamol, because within the 3-4 hours it will get excreted. That is the excretion property of it. How it will absorb, dissolve, and metabolized. Whether the same exists as a molecule, or it is broken down into a simple molecule and it is it. That is one of the very basic and important property for a drug. ADME property.
Okay, thank you, ma'am. The next question. Usually, how much time it takes to analyze a particular sample in HPLC?
How much time? If your sample is ready, you prepared your sample either using a oxalate or using a protein or columnar cryptography. If your sample is ready, based upon the runtime, only thing is you have to preset the HPLC. Like you have to on the switch within a half an hour. You can purge the instrument and you can set up your baseline. Half an hour will take, only maximum half an hour will take for the preparative work. And then, so what is the retention time of your sample? Only 5 minutes and 10 minutes, maximum 30 minutes, right? So within a 5 minutes, you can obtain your chromatograph. My students used to run many 25 samples per day if your program is standardized.
Thank you, ma'am. There is any government virology lab in Tamil Nadu to identify antiviral properties of plant extracts?
Yes. In Tamil Nadu, you have lab. Even as far as my knowledge is concerned, private labs are available. Right? In government lab for virology is King's Institute. You have a King's Institute in Chennai, and also we have Pasteur Institute in Kannur. So they are almost dead. Now only they are rejuvenating. So that is, see how much facility we have without using. Like we have a Pasteur Institute. If you visit in Kannur, it is only 2 hours from Coimbatore. So they are the one, they developed the rabies vaccine, okay? So they have all the facility to produce the vaccine. But still we are depending on others for the vaccine. Only thing our government has put up everything. So they have all the facilities. Now they are setting up the biosafety level 3 lab. I heard now they are rejuvenating the lab in nearby Coimbatore. You have a King's Institute. Only these two. And many private labs are there. But private labs you cannot depend. You cannot trust the people. Like you must be. See, for 15 years back, I contacted one Pune lab to work out on the antiviral drug. That time itself, that professor was saying, "You are a young woman. You have to get a childbirth. Why do you want to work in the virus? It is a need a." Then after hearing all those, I dropped my idea. Okay, now I feel he just wanted to test me whether I am persistently interested. If that time I would have expressed my research, maybe by the time I would have come across with some of the antiviral compounds. So even I left many chances in my life. So rather I have other drugs. I have plenty of anti-cancer drugs in my lab, and which I am not satisfied. Okay. You can approach, or in Pune you have so many labs, in Maharashtra you have so many labs there. In CBS, you can approach.
Now this is question from Ramya. Are you providing outsourcing for testing your samples?
Yes. Actually, I used to do like the thing is like I have only one instrument. Everything is they are bringing the iStem. So you go and browse. Nowadays, analyzing the sample is easy. iStem, there is iStem portal. ISTEM. Okay, they created the common portal that will give you the information regarding where and all the instruments are available, how long it will take to analyze, what is the pricing. Online you can book in online. So they will give the slot and they will give the details, and that even our university is also now going to register under the iStem. And from my personal lab, till now I helped many of the students. I never charge any money. But the thing is, the sample which they are bringing now, sometimes they will lie. So one sample from Bharathiar University, they synthesized nanoparticle with the plant extract. They didn't tell the truth. That time what happened, my column went, and then I struggled a lot to do it. So if you are having a proper sample, I'm ready to help with the HPLC for a few people, not for all. Maybe LCMS, GCMS, it will become a common facility. Definitely within 6 months, because of the COVID, and now they are not encouraging. Sequencing also ready. For HPLC, if anybody if you are in a very critical position that you are not able to get the result, ma'am, from 5 sample, I will help.
Okay, ma'am. Thank you, ma'am. Ma'am, this is from Umera, ma'am. Can you spell it again, ma'am?
What?
Ma'am, actually, we were saying about the portal, no, ma'am.
iStem, iStem, STEM, I step.
STEM. Okay, thank you.
Okay.
I
Ma'am, next
step.
one. Okay, ma'am. Next question. GCMS or LCMS? Which one is better, ma'am?
That's
GC.
again based upon your sample. Okay, based upon your sample. Because if a sample is heat liable, you cannot run it in the GCMS. So if it's GCMS is subjected to a high temperature. But the some sample, like if you want to do a fatty acid analysis, are volatile drugs. Many now volatile drugs are available. Okay, so for that you have to go for the GCMS only. So we cannot say this is best and that is both are equal, only depending upon your sample.
Okay, ma'am. Thank you, ma'am. This is from Joseph, ma'am. Ma'am, shall we bring our students to your lab? Will you permit to
Sure.
visit your lab, ma'am?
With pleasure, with pleasure. I welcome you. Maybe the COVID after the COVID.
It's the last question, ma'am.
Yes.
Whether every component of HPLC has to be used based on the sample to be analyzed, or else only the detector alone to be considered?
No, can you repeat the question?
Yes,
Can
ma'am.
you repeat?
Whether every component of HPLC has to be used.
No. Every component is essential. Every pressure is essential. Okay. Even if you use a proper detector, if you are not setting up correct pressure or correct temperatures or the nanometers, everything is every parameters of HPLC is essential.
Okay, ma'am. Thank you, ma'am. Ma'am, Vera, ma'am.
Yes.
Shall I go to next one, ma'am?
Yes, Prema. If no more questions are there, maybe we can move to the next one.
Okay, ma'am. Now coming to vote of thanks. Gratitude opens the door to the power, the wisdom, the creativity of the wonderful presentation, and for all you have done to spark real interest about the topic. And our sincere thanks to our respected secretary and our dynamic principal, Reverend Sister Dr. Christina Bridget, for their constant encouragement and support. Thank you, sisters. Then I would like to take this opportunity and place a record and our hearty thanks to the entire organizing team for the perfect logistic support and ongoing efforts. I'm also grateful to the technical team for all their efforts, which helped us to witness an uninterrupted session today. My appreciation is not complete without complimenting our enthusiastic participants for their active participation and lively interaction. Once again, I thank you one and all. Thank you. Thank you so much. Okay, ma'am. Thank you, ma'am. Thank you.
I
Thank you
also
so much,
thank
ma'am, for accepting our invitation and spent precious time with us, ma'am.
no, I'm happy to talk with you people because you are my fraternity and which I'm really happy to share my expertise on anything. And also this is the first time I'm just spending 2 hours for the presentation. 1 hour is within my limit. Maybe. Thank you for your interest only. It made me to happen. Thank you very much, the secretary ma'am and who are maybe the head of admin. I thank everyone and I thank Prema who put continuous effort to call me and fix this program. And thank you once again for the opportunity. If you need any help, you can mail me.
Thank you so much, ma'am. Actually, almost the entire 2 hours you occupied with your presentation, and not even a single time you felt disinterested. And it was really a nice presentation, and the way you showed your lab and how to, it was very, very simple and vivid explanation. Thank you so much.
Only because people are bored about the PPTs. Every content is available in Google. If you Google it, you will get everything. So how we can adapt the student only by doing this kind of activity. And
Oh, yeah, that was really nice of you, ma'am. Thank you so much.
thank you.
Thank you, ma'am.
Thank you, Prema. Thank you.
Thank you, ma'am. Thank you so much, ma'am.
Yeah.